Let us now examine about epistasis. Today, scientists realize that Mendel's predictions about the inheritance pattern depended on the genes he chose in his experiments. Mendel carefully selected 7 unlinked genes that control 7 different traits. According to Mendel's predictions, more than one gene controls majority of phenotypes, unlike the phenotypes that Mendel considered. Most of the organisms have more complexity than the characteristics that Mendel studied. Epistasis is one source of this complexity and can occur in different ways, resulting in a variety of different phenotypic ratios. Epistasis is a Greek term meaning above standing or stopping or suppression. Epi means above and stasis means standing. Epistasis describes the state in which a gene masks the phenotypic expression of another. In other words, epistasis is the phenomenon of suppressing or masking the expression of gene by another non-allelic gene. In some cases, a gene at one locus on a chromosome masks or suppresses the expression of gene at another locus and is known as inhibiting genes. These genes inhibit the expression of other genes. Epistatic gene is the gene which suppresses the expression of a non-allelic gene. The gene or locus which is suppressed by the presence of non-allelic gene is termed as hypostatic gene. Epistasis interactions arise because these two genes encode proteins that play a sequence role in biochemical pathway. There are two types of epistasis, dominant and recessive epistasis. If a recessive gene masks or suppresses the expression of a dominant non-allele gene, then it is known as recessive epistasis, while dominant gene masks or suppresses the expression of another non-allelic dominant gene, it is known as dominant epistasis. Now, let us look into dominant epistasis. In dominant epistasis, the dominant gene at one locus suppresses or masks the expression of another gene at a different locus, no matter of its allelic condition, whether it is dominant or recessive. The F2 phenotypic ratio is modified into 12:3:1. For instance, the dominant allele capital A of one gene masks the activity of alleles of another gene capital B and expresses itself phenotypically, then the A gene is said to be epistatic to the gene capital B. That means, the dominant allele capital A can express itself only in the presence of either capital B or small b allele. The alleles of capital B gene will be able to express themselves phenotypically only when capital A gene may contain two recessive alleles small a small a. Therefore, the genotype capital A capital B or capital A capital B small b and capital A small b or capital A small b produce the same phenotype, whereas the genotype small a capital B or capital A capital B small b and A and B all in small produce two additional phenotypes. In this particular table, the mode of action of dominant epistasis is given. In the first column, you can see epistatic alleles. In the second column, it is hypostatic alleles, and in the third column, it is the phenotypic expression of the alleles. If you examine the first row, you can see the epistatic alleles are capital A or capital A small a. The hypostatic alleles are capital B, capital B, capital B, small b, and the third condition is small b, small b, and the phenotypic expression of alleles for these cases are capital A. In the second row, the epistatic alleles are capital A, small a, the hypostatic alleles are capital B, capital B, capital B and small b. The phenotypic expression of the alleles is capital B. And in the third row, you can see the epistatic alleles are capital A, small a, and the hypostatic alleles are capital B, small b, and the phenotypic expression of alleles in this case is capital B. Now, let us examine about the fruit color in summer squash. The botanical name is Cucurbita peppo. Squash is the common name used for 4 species in the genus Cucurbita of the gourd family Cucurbitaceae. Summer squash are a subset of squashes originated in the Americas. It is edible when the rind is tender. Nearly all summer squashes are varieties of Cucurbita peppo, which can vary considerably in color, shape and size. Summer squashes are tendril bearing plants characterized by unisexual flowers, hairy stems and a fleshy fruit of false berry with a leathery rind called a peppo. In North America, the squash are grouped into winter squash or summer squash as well as autumn squash or otherwise known as cheese squash, depending on whether they are harvested as mature fruits or immature fruits. There are 3 types of fruit color in summer squash: yellow, green and white. White color is dominant over other 2 colors and yellow is dominant over green. Yellow color is due to the dominant epistatic gene which is suppressed by its recessive allele W. The color of the fruit is green when the hypostatic gene is also recessive G. In this table, the phenotypes and genotypes of different varieties of summer squashes are given. In the first column, it is the genotype. In the second column, the phenotype is given. If you examine the first row, the genotypic constitution is capital W, small w, capital G, small g. The phenotype is white. In the second row, the genotype is capital W, small g, small g. The phenotype is white. In the third row, the genotype is capital W, capital G, small g, and the phenotype is yellow. In the fourth row, the genotype is small w, small g, and the phenotype is green. When a white colored summer squash variety, wherein the genotype is capital W, capital G, was crossed with a green colored summer squash variety, wherein the genotype is small w, small g, all the F1 progenies are white color and the genotype is capital W, capital G, small g. The F1 plants were self pollinated among themselves. The offsprings resulted in the ratio 12 white, 3 yellow and 1 green in F2. The significance of this dominant epistasis is that the modification of Mendelian dihybrid ratio from 9:3:3:1 to 12:3:1 happens. In summer squash, white colored fruits are produced by a dominant epistatic allele capital W. Another dominant epistatic allele capital Y at another locus produced yellow summer squash and its allele small y for green fruits. Sometimes in this case several genes contribute to the expression of a phenotype as part of a biochemical pathway. According to this pathway, the pigment which is yellow is produced in two steps. The white color of the fruit is due to compound A. The compound A is converted to compound B by the action of enzyme 1. This enzyme is produced only when the plants have a genotype small w. Whenever capital W comes in, the necessary enzyme is not produced. On production of this enzyme, the compound A is converted to compound B, and further the compound B is converted to compound C. The compound B results in green color for the fruit. If the compound B has to get converted to compound C, the enzyme 2 has to play a role, and for the production of this, the plant should have a genotype capital G. If the genetic composition is small g, the enzyme 2 will not be produced. As a result, the compound C will not be synthesized from compound B. Now, let us see about recessive epistasis. In recessive epistasis, the recessive alleles of one gene locus small a mask the action or phenotypic expression of alleles of another gene locus, wherein the genotype can be capital B, capital B small b, or BB alleles. The alleles of capital B locus express themselves only when epistatic locus has the dominant alleles capital A or capital A small a. Due to recessive epistasis, the 9:3:3:1 phenotypic ratio becomes modified into 9:3:4. In this particular table, you can see the recessive epistasis and their mode of action. In the first column, the epistatic alleles are provided. In the second column, hypostatic alleles are given, and in the third column, it is the phenotypic expression of allele. In the first row, the epistatic alleles is capital A small a, the hypostatic alleles could be capital B, capital B small b, small b, small b. Then the phenotypic expression of the allele would be capital A. And in the second row, the epistatic alleles is capital A small a. The hypostatic alleles could be capital B small b. The phenotypic expression of the allele is capital B. And in the third row, you can see the epistatic alleles can be either capital A small a, and the hypostatic alleles capital B small b. The phenotypic expression of allele is capital B. Recessive epistasis in flower color in blue-eyed Mary. The botanical name is Collinsia parviflora. It is a species of flowering plant in the family Plantaginaceae, known by the common names small flowered Collinsia and maiden blue-eyed Mary. When a homozygous white flowered plant was crossed with a homozygous magenta flowered plant in F1, all blue flower colored plants were produced. When the F1 blue flowered plants were crossed among themselves, they produced offsprings in F2 generation as blue, white and magenta flowers in the ratio of 9:4:3. In this table, you can see the recessive epistasis and their mode of action for flower in blue-eyed Mary. In the first column, it is the epistatic alleles are given, second column represents hypostatic alleles, and the third column represents the phenotypic expression, and finally, the F2 phenotypic expression in the fourth column. In the first row, the epistatic alleles is capital W small w, and the hypostatic alleles are capital M, capital M small m, small m small m. In this case, the phenotypic expression of allele is capital W, and the F2 phenotypic ratio is white, and there are 4 progenies. In the second row, the epistatic alleles are capital W, capital W small w. The hypostatic alleles are capital M, capital M small m. The phenotypic expression is capital M, and in the F2 the phenotypic ratio is 3 and it is represented by magenta. In the third row, the epistatic alleles are capital W, capital W small w. The hypostatic allele is capital M small m. Here the phenotypic expression is small m, and in the F2 the phenotypic ratio is 9 represented by blue color. Here complementation results in a wild type F1 heterozygous blue represented by capital W small w, capital M small m. However, in the F2 a modified 9:3:4 phenotypic ratio is produced instead of 9:3:3:1. In this example, the small w allele is epistatic on capital M and small m. On the other hand, capital M and small m can be expressed only in the presence of the allele capital W. In this experiment, the recessive allele is epistatic. This is the case of recessive epistasis. If one or more intermediates in a biochemical pathway are colored, then a different F2 phenotypic ratio is produced. In this example, the enzymatic pathway for the color production from the plant blue-eyed Mary is shown in the figure given. When the blue flowered plants are selfed, the ratio produced is 9:3:3:1, of which 9 offsprings are blue colored. This is because the both enzymes are active in this case. The genotypic composition will have W plus and M plus. Therefore, both enzymes are produced over here, which will finally result in blue coloration of the flowers. In the second row, 3 flowers are magenta colored. Here the genetic constitution is W plus and M plus is absent over here. As a result, enzyme 1 alone is produced, which will culminate in the production of magenta color alone. And in the third case, 3 white flowered plants are produced, wherein the genetic composition is M plus. Therefore, the enzyme 2 is synthesized over here, and there is no production of flower color, and the flower remains as white. And in the last case, only a single white flower is produced, and here no enzymes are produced because W plus as well as M plus are absent. As enzymes are absent, no substrate conversion takes place and the flower color is white.